Cheyenne Bartz with Advanced General Botany here to give you a quick review on photosynthesis, C3, C4, and CAM pathways. Here we can see the light-dependent reactions, but notice over here we have our palisade mesophyll cells that contain chloroplast, as well as our spongy mesophyll cells that contain chloroplast. And that's where we're going to be doing all of our reaction is in these little chloroplasts. Inside we have the stroma space in between, and then the thylakoid grana. Here we've got the thylakoid membrane, and then the lumen is the light colored space on the inside. So here we have the stroma area, the thylakoid membrane, and then the lumen. The main reaction here is that light and water are going to react to form ATP, NADPH, and O2. The AT and NADPH are going to be used for our next reaction. Here we can see the electron from H2O is going up into the photosystem 2. It gets excited by a light and passes through, increasing the photon gradient, gets excited again in photosystem 1 to create the NADPH. And then ATP synthase uses the proton gradient over to build ATP. In the stroma, we have the second reaction, or the light-independent reaction, also called the stroma reaction. Used to also be called the Calvin reaction. I think it even has a second name. But the base of this reaction is we're going to be forming sugars or starches. Here we see that it starts with RUBP, which is a 5-carbon molecule. Rubisco enzyme will fix CO2, but it also fixes O2 to this 5-carbon molecule. When it fixes CO2, we form a 6-carbon molecule that then is broken down into 2 3-carbon molecules. The ATP and the NADPH from the previous reaction is then used to form G3P, which is our main output, and also the reason that we call this the C3 cycle. From there we can see that 2 of those can form glucose, and by adding more of those together we can form more sugars and more starches. The problem with photosynthesis is that it's not a single pathway. It can go both ways. Here we see the main reaction with carbon dioxide, water, forming glucose, and oxygen. However, if you go the other way and react with oxygen and glucose, we do cellular respiration and form water and carbon dioxide, which is the exact opposite of what we want to do. Here down you can see making sugar versus using our sugar. Now we still get a little ATP, so it's not completely horrible. However, it does cause us to not make any sugars. Over here we can see the actual morphology of a C4 leaf is going to be different. We still have mesophyll cells, but the main function is going to be happening over here in the bundle sheath cells. This is called Krebs anatomy, and the stomach of reaction has been moved from the mesophyll cells down to these bundle sheath cells. If you see this little box over here, it's going to be this enlarged diagram over here. Here's our mesophyll cell. And you can see instead of rubisco acting on the CO2, we have PP carboxylase. And that's actually going to form an oxalate, which can then turn into a malate and be transferred through the cell into the bundle sheath cell. From there, it can be broken down into CO2 and pyruvate, which can then be used to make more PP that reacts with the carboxylase and so on. The important thing here is to notice that the Calvin cycle or the stroma cycle, the light independent cycle, I want to make sure you got that, is happening in a completely different cell. So what we can do is actually save all of our carbon and put it closer to the rubisco, which can also act with oxygen. So if the carbon isn't available in CO2, rubisco will use oxygen to form the glucose and go through cellular respiration. What this means is the stomata does not actually have to be open in order for the plant to go through photosynthesis. The plant can open its stomata when it's nicer out, if it's not too hot and the loss of water isn't too great, and collect a little bit of this malate to be transferred over into the bundle sheath cells. And we don't have to worry about rubisco coming into contact with any loose oxygen. And that way, when it's hot and the risk of water is super high, the plant can close its stomata and reduce that loss. The CAM pathway also moves the rubisco away from open stomatas. However, it does this differentiation between night and day rather than between mesophyll cells and bundle sheath cells. During the day, it's warmer and the chances of water loss through open stomata is much greater. During the night, it's cooler and there's less chance of water loss. So what happens with the CAM pathway is that they also use PP carboxylase to fix carbon into malate, which is then stored as malic acid in a vacuole within the same mesophyll cell. During the day, when the stomata is closed, they can use the CO2 from malic acid that is broken down from the vacuoles and then go through the Calvin cycle or the stroma cycle and create sugars and starches from that. And this is all while we have ample light during the day to do the light dependent cycle and have our stomata closed to reduce water. Here you will see C3, C4, and CAM plant pathways. You'll notice that the main difference is the plain stroma cycle in the C3 plant, where rubisco has a chance of also fixating with O2 rather than CO2 and having the plant go through cellular respiration. Here you see C4, where we have the PP carboxylase fixing carbon into malate and then being transferred into the bundle sheath cells, where they will then go through the stroma cycle. On the CAM plant, you see the difference between night and day, but we're in the same exact mesophyll cell. Here, malate is formed by PEP carboxylase as well, and then stored in a vacuole overnight to be used for the stroma cycle during the day to fix the carbon into sugars.